My name is Sarah Kroehler, and today I'll be presenting on my paper for the Journal Club, Microbiome Manipulation by a Soil-Borne Fungal Plant Pathogen Using Effector Proteins. This paper was published by Snelders et al., and it was published in Nature Plants. Why should you care? So, just as host systems have been evolving to fight off infection by pathogens, we also have to recognize that pathogens are developing resistances to this. Evolution is a constant fight for survival, and I believe it's ignorant to think that pathogens are not developing resistances to some of the benefits that we've been discovering that we get from microbiomes. This paper focuses more on effector proteins used by pathogens. And effector proteins have been used to infect and alter host systems, and this has been for a while, and this has been seen in a variety of species. We've seen it in plants due to fungi, in catfish due to bacteria, and in humans by parasites. So this idea is not specific just to plants. However, that is the focus of this paper. The more novel idea of this paper is actually that pathogens can release effector proteins targeting microbiome manipulation. What you usually see in papers focusing on effector proteins is that they're more looking at host interaction. So this paper takes a different approach and looks at how it affects microbiomes. So there's a couple terms to know. The first is effector protein, which is the bigger focus of this paper. Effector proteins are small cytosine-rich proteins with various functions. A couple of those include manipulation of host immune responses, self-defense of the pathogen, or the acquisition of nutrients from host tissues. These effector proteins can be used through various secretion systems. They can be secreted just into the environment. So there's a lot of different ways effector proteins can be used, but the idea is that they're proteins with effects, basically. The next term is antimicrobial peptides, or AMPs. And these are proteins with specific microbial properties that are based on their secondary structures. So it's how the protein folds. AMPs are typically pretty conserved. However, mutations such as SNPs, indels, all of that are very likely to change their function due to the nature of it being due to its secondary structure, the way they fold. So mutations can cause differences pretty easily. The next two are more microbiology based, more related to our class as a whole. The first is the principal coordinate assay, and this takes a dissimilarity matrix and creates a visual representation of the differences between two data sets. This is a really good way to visualize relationships, and it's pretty commonly used in microbiome papers to show differences in diversity. The other one is the pairwise bacterial order comparison, and this is a technique that's used, or an analysis that's used to understand relationships and interactions between groups such as orders or taxa, as opposed to individual species or strains. And this gives us a better idea of the relationship within a complex microbial community, like as a whole. They used a couple different molecular techniques in this paper. The first one is real-time PCR. The second, as this paper is focusing on effector proteins, is biorad protein assays. And this is used for just measuring different protein concentrations. They also use two different sequencing techniques. The first is 16S amplicon RNA sequencing through Sanger sequencing. They use this for a couple of the bacterial isolates. Others, they gathered the sequencing from various databases. I'm not 100% sure why they chose to use Sanger sequencing for this, but my best guess is that they only used it on 6 bacterial strains. So they chose Sanger sequencing for its higher accuracy as opposed to its speed. The other type of sequencing that they used was Illumina sequencing for the root microbiome genomic DNA sequencing. And I believe that this was a better choice due to how high throughput Illumina sequencing is, as root microbiomes have a lot of species and a lot of DNA to be sequenced. I believe this was a better choice. In this paper, we'll be focusing on a soil-borne fungus known as Verticillum dahliae. This fungus is known to cause vascular wilt disease in a variety of plant species. It is a soil-borne fungus, so that means that it invades host plants through their root systems. So the specific plant microbiome that we will be looking at will be the root microbiome. And they actually had already determined in a previous paper that the effector protein we'll be looking at, which is VDAV1, they had already determined that it was a virulence effector protein. We already knew that before going into this paper. So this paper was a transition into seeing how it affected the actual microbiome of the plant. So the first thing that they wanted to do was to actually determine what the antimicrobial activity of the effector protein was. And they did this by taking a couple different plant-associated bacterial strains, and they incubated it either with or without the protein. You'll see that there are 3 lines. The red line in this graph is MQ. That's just demineralized water, and it was used as a negative control. We see the green line is the VDAV1, our effector protein that we're looking at. And they also included the blue line, which is VNAV1. And what VNAV1 is that it's actually a pretty close homologue to VNAV1, only it comes from a different fungal species, which is Verticillum nubilium. So just a different fungal species, but a very closely homologous protein. And so when they incubated these bacterial species with the isolated proteins, they saw a variety of effect. The first one, what I have highlighted up now, are all of the gram-positive bacterial species. What you see when you look at these 4 graphs is that the VDAV1 actually reduced the activity of all of the gram-positive bacteria. They all had inhibited activity. However, when we look at the other bacterial species, which are our gram-negatives, there was a much wider range of effect. Some examples that they gave were between these two, Acidovorax and Ralstonia, which are actually species within the same order, but you can see that in the Acidovorax species, it actually had a greater effect. Another example is these two, which are 2 closely related rhizobials. And you see that the rhizobium species had a much greater, there was a much greater impact on the rhizobium species as opposed to tumefacients. So we see that the gram-negative was not necessarily less susceptible, but the different species had various sensitivities to this effector protein. Unpictured in this graph, they also did this same experiment with different fungal species and with plant protoplasts, which are just plant cells without the cell wall. It makes them a little more susceptible to infection. And they found that there was no antifungal or phytotoxic activity, meaning that this effector protein is specifically for bacterial species. The reason that they included our 3rd line, our blue line, from the V. nubilium was to find out how V. dahliae actually acquired this. And they found that it only inhibited a subset of the bacteria that VDAV did, or it inhibited them to a smaller effect. This indicated that the antimicrobial properties are likely not from horizontal gene transfer from a different fungal species. It was either a random mutation or something acquired from plants or the microbiome, but it was not something that was acquired from other fungal species. Knowing that there was antimicrobial activity from VDAV1, they next wanted to determine if it was bactericidal or bacteriostatic. They wanted to know if it was killing or just inhibiting growth from these bacterial species. So what they did was they exposed B. subtilis, which was highly susceptible to VDAV1. They exposed it to the protein, and then they used electron microscopy to see what it was doing. You'll see that over time, they saw blebbing, swelling, and lysis in the bacteria. And this implies that the effects are actually bactericidal. It was killing this effector protein was killing the bacteria. So they proposed that V. daliae actually uses its effector protein to affect microbiome composition through the suppression of other competing microbes. They did bacterial community analysis using 16S ribosomal DNA profiling on both tomato and cotton root microbiomes. As I said, we know that V. daliae causes diseases in a lot of plants, so they chose two to look at. They used the wild type V. daliae, and they used a VDAV1 deletion mutant to compare how the effector protein works. This was conducted at 10 days post-inoculation because they wanted to try to minimize any effects from disease symptomology. As the disease progressed, they didn't want to affect the bacterial species from that. So what we have here on the top row are all of the tomato, and the bottom row is all the cotton, just for reference. The principal coordinate analysis, the PCA, showed clear differences between the root microbiomes of the wild type and the deletion mutant. The wild type you can see in blue, and the deletion mutant is in red. So we can see that there is clear separation in both of the plant types, in both tomato and cotton. So the root microbiomes are different when the effector protein is present. They then did pairwise bacterial order comparisons to look at the differential abundances between the two treatments. For tomato, on the top row, they found 3 different orders, and what they were most interested in was the Sphingogomedalis, which is on the top, as it had the greatest decrease, meaning that it had in the wild type, so it had the greatest sensitivity to the effector protein. Cotton was a little more complicated. There wasn't a huge difference when looking at the order level. So they did a agglomeration of amplicon sequence variants based on phylogenetic relatedness. They basically dropped it down to the family level, which revealed 8 taxa, with one of them still being Sphingomonadaceae, which falls under the Sphingomonadellis order. This one was also seen to be the most different when treated with the wild type, again pointing to its increased sensitivity when compared to the other bacterial species. In fact, they found it to be consistently and completely absent when they tested the wild type. They didn't find it in there at all. Knowing, like I said, they were really focused on the Sphingomonadellis order. So they wanted to test which were the most sensitive, and they compared the average relative abundances of the 2 treatments. And they found that 4 genus were the most affected by this, and these are known as the sphingomonads. You see that in group 3, and the different genus have various effects, but they found a couple different ones that were highly affected. The next thing that they wanted to do was they wanted to confirm that this was a direct result actually, of VDAV1. They wanted to make sure that they weren't seeing this as a result of it being implanta, that the host plants weren't having effects. So they created a synthetic community and treated it with the purified protein. Once again, we see that they used water as a control when you look at D. And it once again showed that there were clear differences in the composition of the bacterial community when it was treated with the protein. This again confirmed the impact on Sphingomonadellis in planta and that it was a direct result of the VD-AV1 activity. So next they wanted to see, next they did transcriptional analysis, which actually showed that VD-AV1 is active during growth conditions that mimic soil colonization. This means that VD-AV1 is not only transcriptionally active when it's actually infecting the plant. This suggests that VD-AV1 actually helps facilitate colonization within the soil as well. So they did colonization assays conducted in various soils and mediums, which is what we see here in figure F. So you'll see that what we really want to look at for these is the untreated versus the sterilized, basically, and the without soil. So the without soil, while not sterilized, does not have the bacteria in it. It's just on a growth medium. So when we're looking at the untreated versus the rest of them, we can see that those are the only two that have clear differences in the V. Dalliae biomass, which is how they measured how the effector protein contributed to colonization. They wanted to see how much the fungus grew in these soil conditions. And it demonstrated that in the presence of the normal soil microbiota, it actually contributed to the fitness. The increased biomass that we're seeing is the increased fitness. And these differences were not detected in the sterilized soil or the MS without soil, as these don't have the normal soil microbiome, plant microbiome that we know VD-AV1 affects. So we're not seeing its contribution to V. Dalliae colonization. All this to say, this is showing that the contribution of VD-AV1 on the growth of V. Dalliae is due to its antimicrobial effects. We only see the effect when we have the microbes present. They wanted to, knowing that this is due to the antimicrobial activity and knowing how interested the researchers were in these sphingomonads, they wanted to see how sensitive different species of them actually were. So they took 6 plant-associated sphingomonads and they incubated them in vitro with the purified protein. And as you can see from figure A, this agreed with the earlier pairwise bacterial order comparison, and all 6 species were found to have inhibited growth when treated with VD-AV1. Because all 6 had decreased growth, this indicates that the order itself has broad sensitivity. It affects most, if not all of them. So they hypothesized that in the absence of VD-AV1, these species may actually act as antagonists to V. Dalliae. So the broad sensitivity kind of implies that this specific protein was actually created as a response to these bacteria, as a specific way to fight this specific type of bacteria. They wanted to test this, so they grew V. Dalliae with 2 different species of the sphingomonads, and they used the wild type and the deletion mutant. And they found that when looking at figure B, they found that the deletion mutant in the middle had reduced biomass. So what this means is without the effector protein, they saw that the bacterial species was actually decreasing the biomass of the fungus. This shows that the bacterial species is most likely an antagonist to the B. dahliae colonization. Continuing on this, they wanted to see how much more VDAV1 contributed to host fitness and host colonization as a response to these to this bacterial species. So they did a treatment of sterilized tomato seeds with S. macrogoltabeta, which is a species of the sphingomynads. And they actually found, when you look at figure C and figure D, these are the ones most important to this, you actually find that there was a decrease in the progression of the verticillum wilt disease, the disease caused by this fungus. And the plants in figure C line up with figure D. I know it's slightly offset, but you see the middle ones, which are the smaller, more stunted growth, are actually the wild type species. So the wild type species has the VDAV1 protein, is able to fight off the sphingomynad species, and therefore we see a progression of the disease, basically. But in the deletion mutant, what we see is without the effector protein, the normal plant bacterial species is actually able to not necessarily, but slow the disease progression. The plant's a little bigger. It looks a little bit healthier. There's more leaves on it. So what the researchers took from this is that the sphingomynad species actually has plant protective effects. So when the sphingomynad species isn't wiped out by the fungal species, it actually helps to protect the plant from the fungus. They then did quantification of this data by taking the B. dahliae biomass and the relative abundance of the sphingomynads, and they compared the two in the remaining figures, E, F, and G. And you'll see that VDAV's secretion significantly affects its proliferation to counteract the positive effects. That's a little confusing, but you'll see that with the VDAV deletion mutant, the relative abundance of the sphingomynads was higher. So no protein, more bacteria. And then you compare it to the biomass, and you'll find that a higher amount of the bacteria leads to a lower amount of biomass. This supports the hypothesis that V. dahliae is secreting the effector protein, VDAV1, specifically to target antagonistic bacteria during the host colonization. The one caveat that researchers really had is that they were not able to test this on a germ-free line of tomato plants. They were unable to establish this, but they understand that this is a shortcoming of this experiment. So, knowing that VDAV1 exists to fight against antagonistic bacterial species, they wanted to see if there were any other effector proteins that were also around to suppress microbial competitors. Through sequencing, they found 10 candidates that were structural homologs, but were not seen expressing during host colonization. So they had 10 candidates, and when they colonized a host, they did RNA sequencing, and they didn't find any to be expressed. So the thought was that in vitro colonization, just didn't induce any of the effector genes, and that they would need extra environmental triggers to actually induce the expression of these. So they then took this they then took the colonization and did it in a soil extract instead of in vitro, and they found that in soil extract there was induced expression of VD-AMP2. You'll see in figure A that in soil extract they actually found expression, but in the media in PDB they did not. So VD-AMP2 is another effector protein that has about 90% structural similarity to VD-AV1. They wanted to test the antimicrobial activity of VD-AMP2. However, they had a hard time growing or producing a purified version of this protein. Usually this is done in bacterial species, and they were having a hard time getting the species to grow and produce this protein. So what they decided to do was to take VD-AMP2 in the V-Dalliae, and they altered the V-Dalliae to put it under the control of the VD-AV1 promoter. So that means that under the conditions that VD-AV1 would normally express, VD-AMP2 would express now. And they took a V-Dalliae wild type and the V-Dalliae VD-AMP2 mutant, and they cultured these with bacterial species, and they did find antimicrobial effects in B. subtilis and P. corrugata. What they didn't show in this figure is they did it in a couple other species and didn't really see any effects, including the Sphingomonidalis. So what this suggested is that this effector protein did have antimicrobial activity similar to VD-AV1, but they had different activity spectrums. This would imply that VD-AMP2 is an effector protein that evolved to target different bacterial species than VD-AV1. They then wanted to see how this effector protein may contribute to V-Dalliae fitness, whether it was created to counteract antagonistic bacteria. So they expressed this fungal mutant with the VD-AMP2 in different soil types, with and without microbes in it. And they saw that in untreated soil, if it didn't express VD-AMP2, if it did express the VD-AMP2, it decreased the biomass. But I'm sorry, I totally read that wrong. So they saw that there was an increase in biomass in soil colonization assays, which shows its effect on the V-Dalliae fitness. And they saw that there was a increase in the V-Dalliae biomass when you compared the re-inoculated soil compared to its sterilized soil. And this once again points that the contribution to the biomass is due to the antimicrobial properties. So now we're starting to wrap up. So as it becomes more accepted that the host microbiome contributes to its ability to suppress disease development, we also have to understand how disease-causing agents are developing to fight off bacterial antagonists. This paper specifically shows evidence of effector protein activity that's targeting not the host directly, but the microbiome and manipulating that. Researchers hypothesize that as we've already found two effector proteins with this function, that there are likely others with similar functions that are just triggered by different environmental factors, different bacterial species, different soil compositions. And they also think that some of these are potentially to fight off opportunistic microbes when host microbiomes have been impaired in later disease stages. They finish off the paper by hypothesizing that future research into these effector proteins, what microbes they target, and all that may actually lead to the discovery and development of new antibiotics. If we look at effector proteins specifically through the lens of how they kill or inhibit bacterial growth, we might actually discover some new antibiotics. And finally, I have a couple questions to try and start the discussion. If this is prevalent in plants and fungi, do you believe that this is an area of research that we should expand to human microbiomes? While it may already exist, I'm not positive. Should we expand on how much research we're doing in this area? The second is, knowing that microbes are constantly evolving, what do you believe the next step for bacterial species in the microbiome is going to be against these bactericidal effector proteins? How do you believe the bacterial species are going to evolve to combat this? The third is, do you believe that this research has significance for the agricultural sector as a way to decrease disease prevalence? Is this a way to target bacterial species or something to target against fungal species? Do you believe that there's a profit to be made for the agricultural sector? The fourth one is, do you believe that this research can actually be used to create new antibiotics, or is that just a lofty goal? We haven't had new classes of antibiotics come out in a couple of decades, and do you believe that the researchers are right that this could be a new way to find them? And the last one is, if antibiotics are created from this research, do you think that they would disproportionately target our beneficial microbes? Do you believe that the way that these effector proteins start out targeting microbiomes, do you believe that they might actually target our beneficial ones more than they would target the ones we want to clear? And that concludes my presentation. Here's a list of my references. Thank you.